The document published February 29, 2024, Evaluation of Oxidative Stress and Genetic Instability Among Residents Near Mobile Phone Base Stations in Germany, explores the potential health impacts of long-term exposure to radiofrequency electromagnetic fields, RF-EMF, emitted from mobile phone base stations. The study assesses oxidative stress, DNA damage, chromosomal aberrations, and cancer-related genetic markers in individuals living in proximity to these base stations. With a focus on non-thermal RF-EMF effects, the research finds higher levels of lipid oxidation and oxidative DNA lesions in the exposed group, although not statistically significant. However, significant increases in chromosomal aberrations such as dicentric chromosomes, chromatid gaps, and chromosomal fragments were observed in those exposed to higher RF-EMF levels. The study contributes to the understanding of the potential genotoxic effects of chronic exposure to low-dose RF-EMF, highlighting the need for further research in this area. The document provides a detailed examination of the effects of radiofrequency electromagnetic radiation, RF-EMF, emitted from mobile phone base stations on oxidative stress and DNA damage. It outlines various methodologies and findings from the study, offering insights into the biological impacts of chronic RF-EMF exposure. Here are the key points regarding the proof that RF-EMF exposure can cause oxidative stress, and the support for these findings. Increased levels of lipid peroxidation and oxidative DNA lesions. The study found higher levels of lipid peroxidation and oxidative DNA lesions in the exposed group compared to the control group, although these differences were not statistically significant. This suggests that RF-EMF exposure may lead to oxidative stress, a condition characterized by an imbalance between the production of reactive oxygen species, ROS, and antioxidant defenses. Significant chromosomal aberrations. There were significant increases in chromosomal aberrations such as dicentric chromosomes, chromatid gaps, and chromosomal fragments in individuals exposed to higher levels of RF-EMF. These findings indicate that chronic exposure to RF-EMF can lead to genetic instability, which is a hallmark of oxidative stress and its damaging effects on DNA. Comparative analysis with other studies. The findings of this study are supported by other research that describes the non-thermal effects of RF-EMF exposure, including oxidative stress. For instance, oxidative mechanisms have been proposed to explain the biological activity of low-intensity RF-EMF radiation. Studies have shown that exposure to RF-EMF can induce oxidative stress, leading to damage of macromolecules, inflammation, and accumulation of mutations, which are biomarkers of elevated cancer risk. Watch Part 2. Methodology for detecting oxidative stress and DNA damage, or visit www.rfsafe.com to learn about oxidative stress and DNA damage from cell phone radiation. Be RF safe.